Chapter 3, Section 12, Autotrophy and Nitrogen Fixation. Okay, now we're going to be looking at, up till now we've been talking a lot about catabolic, taking organic or inorganic compounds and breaking them down in the process of consuming them, liberating energy in the form of excited electrons, ATP synthesis, and all of that fun stuff. Well, now you have a bunch of ATP. What are you going to do with it? You're going to have to build stuff. You're going to have to replace, replenish. That's what anabolism is. Anabolic metabolism. Taking small things, linking them together to make bigger molecules linked together to make macromolecules. You're going to have carbon and nitrogen here of some form. Okay? At the basic level, many microorganisms can take carbon dioxide as well as nitrogen gas from the environment, from the atmosphere. They can take it and do what is referred to as carbon dioxide fixation or nitrogen fixation. This means they can string together individual carbons from carbon dioxide to start forming 2 and 3 and 4 carbon strands, which can then act as the precursor for building other things. Remember a few sections ago when we talked acetate. Nitrogen fixation means you're going to take gaseous nitrogen and you're going to start linking it together with hydrogens and other things to start making things like ammonia and urea and nitrite, which is now, instead of a gas, it's now in a solid form, which can then be added to carbon-containing compounds, make amino acids, make nitrogenous bases for nucleic acids. This is a very energy-dependent process, whether you're talking CO2 fixation or nitrogen fixation. You've seen some of this before when you're talking photosynthesis and intracellular or plant biology or cell biology. The Calvin cycle is a great example of this. Autotrophs, organisms that can make their own carbon molecules, their own organic molecules using gaseous carbon dioxide. Bacteria, purple bacteria, cyano, algae, green plants, chemolithotrophs, few archaea. Notice no mammals, no animals, no fungi here for this. Requires CO2, some form of an acceptor of CO2, lots of NADPH, lots of ATP. You're going to have Rubisco there. You're going to have a sugar to build on. And then phosphoribulylkinase. First thing you do is you're going to take Rubisco. And you're going to add on some CO2. You're going to take some pre-existing some pre-existing sugars, short chain sugars, and you're going to start adding on carbon dioxide to each one. You're going to go through, paste these together, rearrange them until you get glyceraldehyde 3-phosphate. Hey, G3P, remember that was the midpoint in glycolysis. Take some G3Ps, link them together, you got glucose, the reverse of glycolysis. So you're taking Rubisco, ribulose bisphosphate, 1, 2, 3, 4, 5. And you're going to start doing stuff to this. You cut it in half. 1, 2, 1, 2, 3. I'm sorry, 1, 2, 3 carbons. 1, 2, 3 carbons. 1, 2, 3, 4, 5. There's my carbon dioxide. So you have 6 total. It's an unstable intermediate, so it's going to get cut in half. You now have 2 3s. The 2 3s with some energy, going to get reformed. Things moved around. Boom, boom, boom. Rearrange the deck chairs. PGA, 3-phosphoglycerate, becomes 1,3-bisphosphoglycerate, which becomes glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can be linked together to form glucose. Can be fed into the citric acid cycle, or broken down and made pyruvate to feed into, do whatever with. Your textbook goes and talks about, considering it as taking 6 carbon dioxides, link them together to make 1 hexose. Hexose is another way to say glucose. Hex, 6 carbons. This means this was probably somebody with a really strong biochemistry background. If it's hexose and they say hexose, that means chemistry thinking, biochemist. If it was a biologist, microbiologist thinking this, they would have just said glucose. Tomato, tomato. Basically C6H12O6, hexose glucose, however you want to think about it. The ribulose bisphosphate, 6 of those, and 6 carbon dioxides are required to keep this process going. This will result in 6 of the ribulose 5-phosphates, 30 carbons, and 1 6-carbon thing, or 2 glyceraldehyde 3-phosphates kicked out. So this is a very, very energy dependent and lots of moving parts dependent. You're not just taking a bunch of carbon dioxides linking together to make 1 sugar. No, you're taking carbon dioxides, you're adding them to a pre-existing molecule, rearranging that molecule so you can kick out a sugar. And then you have to go through many, many steps to take what's still there, your leftovers, the majority of what you started with, and rebuild, regenerate what you had before. I don't like these numbers, okay, because these numbers don't match up with what you're going to see in a lot of different textbooks. They're saying 6 ribulose, also known as rubisco, 6 carbon dioxides, go through, rearrange. Yes, the numbers add up, but it's not what we taught you in Intro Cell and Molecular. It's not what we teach you in cell. It's not what we teach you in plant biology. 6 carbon dioxide, so there's 6 carbons. Ribulose, it's a 5-carbon thing, so that's 30 total carbons. Add those all together, and then go through and cut, paste, move stuff about, and you now have 12 3-phosphoglycerates. Okay. Add in some energy. You now have 12 1,3-bisphosphoglycerates. 12 3-carbon strands. Still 36 carbons. Add in 12 hydrogens. You now have 12 3-carbon glyceraldehyde 3-phosphates. You remove 2 of those, 2 G3Ps, so that 6 have been removed, 10 left. So 2 of these G3Ps get taken out, you still have 10, 30. This is that regeneration part. You need to go from 10 3s to 6 5s. 6 times 5 is 30, 10 times 3 is 30. So all of this is going from glyceraldehyde 3-phosphate back to our ribulose. Fixation, reduction, regeneration. It's just the numbers are going to be different than what you've seen in other textbooks, other courses. Nitrogen fixation is something that's quite a bit different. Okay? Nitrogen. Think about all the compounds, all the organic molecules in your body that have nitrogen in them. All amino acids making up your proteins, the nitrogenous bases making up all your nucleotides, and then there's numerous other organic molecules throughout there that have, a nitrogen-containing functional group on them. The majority of this all starts with microbes. Microbes will take nitrogen, gaseous nitrogen, and will fix it in the form of NH3, ammonia, or NO3, nitrate. All starts with gaseous nitrogen. In the process of doing this, these microbes will overmake either the ammonia or the nitrate, the nitrite, and then they will excrete it out. If they are soil microbes, they excrete it out into the soil. Hey, guess what is very dependent upon nitrogen, either in the nitrate or the ammonia or the nitrite form? Plants. Plants will then absorb all that excreted, fixed nitrogen. They need it, they will absorb it through their roots. It gets into the plants, it becomes part of the plants, proteins, nucleic acids, or other organics. We being animals, come along, eat plants. There's where we get our nitrogen. It's so cool. So when you look at this, fixation, when you look here, electrons for nitrogenase, nitrogenase activity. N2, gaseous nitrogen, gets fixed with hydrogen, so you have N2H2. Add in 2 more, so you split this, add in some more, and you go from gaseous nitrogen to 2 NH3s, 2 ammonia. In the process of doing this, you're taking things like pyruvate, turning it acetyl-CoA, releasing carbon dioxides. Flavodoxins, taking those excited electrons and utilizing these to go through and reduce dinitrogen reductases. Dinitrogen reductases are ATP driven. They will go through and they will take dinitrogenase, boom, boom. In the process, energy is being transferred from one chemical compound to another, so that ultimately N2 becomes 2 NH3s. Oxidized, reduced. Oxidized, reduced. Flavodoxin gets reduced due to pyruvate becoming acetyl-CoA in these organisms. The reduced flavodoxin will now reduce dinitrogen reductases, which will then be phosphorylated, and in the process, dinitrogenase becomes reduced itself because, boom, dinitrogen reductase reduces it. This enzyme reduces this, and then this dinitrogenase takes dinitrogen, nitrogen gas, and will reduce them. So this becomes oxidized. In the process of oxidizing dinitrogenase, you reduce dinitrogen, N2 gas. Now becomes NH3. Nitrogenase is an enzyme complex. You have dinitrogenase, dinitrogenase reductase. In the process, you're using some iron molybdenum cofactors. Remember, cofactors help enzymes do their job. Cofactor will help either in the active site or get the enzyme to the correct shape so that it has an active site. Some electron donor is going to pass on its electron to dinitrogen reductase. Dinitrogen reductase is going to reduce dinitrogenase, which then ultimately reduces what they're calling dinitrogen, which is basically N2 gas. In the process, you need a handful of electrons, because each of these steps is going to be excited electron driven. And a whole lot of ATPs. This is not a cheap, an energetically cheap process. It's inhibited by oxygen. So nitrogen fixers are generally going to be anaerobic or grow best in anaerobic conditions. If it's an obligate aerobe, this process, the nitrogen fixation, is protected from the oxygen, because remember, oxygen is highly reactive. Oxygen will strip off electrons, strip off hydrogens from a lot of things it comes into contact with by slime layers. So some of those bacteria that have a slime coating will localize their nitrogenase and all these complexes we just talked about in their slime layer, because it'll be oxygen poor in that layer, and it'll be a great place to fix dinitrogen. You can see this with these heterocysts. This slime layer out here is going to be rich in nitrogen fixing compounds, nitrogen fixing enzymes. Not here in the cytoplasm, where it's going to be oxygen rich, because the oxygen will pass through, readily pass through, and it will not interact very well with compounds, components that are stuck here in the slime.